Kip Sweeny. It's good to have you here. April 13th, 2011. Today we're going to discuss an important information about making money. How many people here would like to make $1 million a year? Oh yeah. Well, if we take this plant right here and we use small cells of this plant here, we can make about 1 million plants from these, sell them for $1 a piece to any of the box stores. Have you ever walked into one of the box stores and you see all the plants, they look the same height, same width, about the same color? A lot of those are cloned plants. We call that microbiology. I'm going to go over this information right here. Micropropagation, propagation of plants through tissue culture, and some people will recognize it as cloning the plants. You'll notice right here, tissue culture is the ability to establish and maintain plant organs of leaves, shoots, and roots in an aseptic environment. Can anybody tell me what aseptic is? Yes, ma'am. Sterile, exactly. The key to success in tissue culture and cloning plants is the sterile environment. If you can get a sterile environment, then we will have nothing to interfere with that one little tiny cell becoming a plant with roots, stems, and leaves. If you'll notice right here, an aseptic procedure, we ensure that the cultures are free of microbes that can overwhelm and kill a tissue culture explant. The explant will actually be the plant that we're trying to grow. Yeast, fungi, and bacteria. All those items are on our bodies all the time. And if you get into an aseptic environment, we want to keep all those items out. What I'm going to do is I'm going to show you how to do that today. We're going to practice it here, and then we're going to move into the lab, and you'll be able to do this yourself. The tissue culture hood is what's going to keep the environment sterile. If you'll notice, the laminar flow cabinet or the laminar flow closet is carefully enclosed bench designed to prevent contamination. Here's a picture of that right here. If you notice her workstation, it's a sterile environment. If you ever look on top of your stove at home, you have a fan that's got a vent and a light that's blowing air or sucking air. Well, imagine that warm air is being blown across the table to me so I don't contaminate the environment. You'll see that in the lab when we get there. What I want to do is I want to show that to you. So we're going to keep moving. There's 4 stages of tissue culture or cloning the plants. Stage 1 is the establishment of the plant in the petri dish, which we'll show in a moment. Stage 2 is multiplication by dividing it and dividing it and dividing it. Stage 3, we want to get roots to grow into the medium, and then we'll transplant it into soil, and then we'll acclimate it by putting it in a greenhouse. Are you ready? Yeah. All right. We're using a substance. This is called agar. If you notice right here in these dishes, this right here is the medium. Instead of using dirt or soil, we're going to use agar. We placed them inside the petri dish. We have a sterile environment. I'm going to take just 70% rubbing alcohol or 10% of bleach, and I'm going to sterilize my hands because these are the items I'm going to use. And I'm going to take my utensils that I'm going to work with, and I'm going to sterilize them. When I go into the environment here that's sterile, it's now clean. I open up my petri dish. The petri dish has the agar in it. I'm going to find my specimen. We'll say right here, how many of you like strawberries? Lots of strawberries. We're going to take the item, place it in there for just a few moments, and sterilize it. We're going to lay it on the table, and then we're going to go ahead and just cut tiny little pieces. Can you see that tiny little piece? We're going to place it right smack in the middle of the agar. We close the lid. It's now a sterile environment. Within a matter of 42 hours to 72 hours, if there was any contaminants of fungus, bacteria, or mold, it will grow. But if it doesn't, if it stays clean and all you see is that little green speck, then within weeks you'll see roots grow out, stems grow out, leaves grow out. We're going to take this bad boy by using some stretch tape, and we're going to enclose it to ensure that the environment stays safe. You can try this when we get into the lab, just taking the stretchy tape and moving around the petri dish as such. Like I said, in about 6 weeks, that's going to be a plant just like this, with roots, stems, and leaves. If I can sell this one for $4.99 at the box plant, and I sell 1,000 of these, did I make some money? So this is why we want you to get involved in tissue culture, cloning plants, and microbiology. Does anybody have any questions? All right, well, we're going to go ahead and we're going to cut it at this moment, and we're going to move into the lab, and you're going to do this for yourself. Thank you.